Let me try checking there.
Every work has been marked. Celtic, have you received your results? Okay, Maria, can you be able to hear me? Okay, now,
Master, mine is not there yet.
Open the question. You mean it is not yet marked?
Yeah, it is not there.
I don't know what you mean by it's not there, but I think, let me try to check because, and I diagnose the problem very fast. Because
I've seen it.
And is it marked?
Yes.
And what did you get?
10 out of 15.
Okay, let me try to open some of your work and then I show you some of the mistakes which you did, because I would expect you to get I would expect each one. Yeah, I'm seeing some people have just handed in because I had marked every work. Okay. Most of these people, I think you are seeing, they have just handed in. Here.
Yeah, good day. It's always
Okay. Okay, I think so let me go straight to making a few comments of how the people performed. People are still, I think they are still Hilary, do you have questions as far as the marking was concerned?
not really.
Okay, now let me try to review for you. You are supposed to describe the structure of the stomata. If you did remember, you did the one of the thickenings. try and go back. I want to first comment about the structure of the xylem. Okay, just a moment. Yeah. So I think there is a thickening which you are confusing. Multiple spiral thickening, you are confusing it with you are confusing it with scalariform articulate because it forms those kind of network or ladder-like structures.
Yes.
Different the max because of that thickening, and then the adaptations. I think the adaptations you're able to get.
This one.
Where we looked at the main conducting. Okay, you were looking at maybe there is one which you confused. There is one whereby you say that the vessel elements have end perforations in the end walls, and then you say the tracheids don't have perforations.
Yes.
Okay, so you don't compare something which, for example, the vessel elements, some of them have end walls, and these end walls, and some don't have. So you wouldn't want to compare such a scenario with this one. And maybe that's what I wanted to comment on from your adaptation that you are giving.
Okay.
Then, since I've been able to show you, let's just go straight to, and then I show you where this topic ends. Because I'm seeing many people, since they did their exams, so they think it was over. And I think previously we had discussed
mass flow.
Mass flow, yes. We had finished mass flow hypothesis, and we're supposed to look at cytoplasmic streaming. Now, to make you people a little bit, so after mass flow, we're supposed to look at cytoplasmic streaming, and then we go to electroosmosis, then we go to surface spreading, loading and unloading. This one is well explained in biological science. That is the same thing. Then mineral elements are necessary for plant growth, where we have macro and micro. And then looking at special methods by which plants obtain essential nutrients, whereby some roots possess mycorrhizae. I don't know whether you've heard of the word mycorrhizae before.
No.
Okay, myco comes from mycology, which is the study of fungi. So myco, that is it has to do with fungi. Then rhizae from rhizoids, which means roots. So fungi with roots. So mycorrhizae is an association between the fungus and the roots. And these ones will, what actually this association is that both benefit, whereby the fungus digests or breaks down the humus in soil to release nutrients, and then they are absorbed by the roots, and then normally they get attached onto the roots form of support. So that one is very common, and they normally examine it, as well as root nodules. I don't know whether they've seen swellings on roots, especially of brown nuts.
Yes.
Those are called root nodules. And then we also talk of the carnivorous plants. Under carnivorous plants, we talk of the pitcher plant, this one, and the Venus flytrap. These ones trap insects and yes.
Are those the plants which capture insects to get nitrogen.
Clear all. Can you please repeat the question, because I didn't get your question since the network was unstable. Hillary, your network is unstable. I can't hear you. Hillary, can you hear me? I can't get any feedback from you. Hillary, can you hear me? With this lesson, since even the one learner which we have can you hear me? Can you hear me?
Hello, Mr. Patrick.
Yes, can you hear me very well?
Yes.
You were still asking a question and the network disturbed you.
I say that is the plants which capture insects to get nitrogen.
That is very true. Because they normally grow from soils which lack nitrogen in them.
Okay.
So they are the very ones. Okay, and then going forward, yeah, so this topic was supposed to end here. Of course, we have spent a lot of time, since normally our lessons have been having just 1 hour. And that's why we went slowly and slowly and slowly. So at this moment,
Yes,
I would like you to, if you have questions concerning biology, I may throw more light, because this topic is done. And yeah, do you have questions concerning biology? And we
last time I have a question. I want you to guide me in paper 2, especially paper 2. How best can I achieve more in paper 2?
Okay. Paper 2 is about writing. It's about writing a lot.
Yes.
And how can you, like, many students fear practicing, especially graph numbers and writing. So that's where the challenge comes in. So a student is comfortable to just read and then stop there, or just answering a few objective questions or a few structured questions. But making them to sit down without that is copying, because many students, they will prefer attempting paper 2 questions which have answers already. That is in they call them pamphlets, which have answers. And then they attempt a question, then when they are stuck, they look at the answer and like that. But if you can get that two questions from the topics.
Yes.
Don't read topics which you've not yet done. You get topics which you have covered. Is you sit down.
Yes.
answer what you know. Uh, and you have to go on and use textbooks to generate answers.
Yes, master.
Yeah, you use the textbook and then you generate the answers. So that is one way of how you can go about paper two.
Okay.
And then you must, because number one is normally which has more answers, 40 marks, more marks. So you must be comfortable with one, first of all, plotting the graph.
Yes.
After plotting, you should be able to describe the graph. And then after description, you should be able to explain different ways of how it is examined. Normally using words like accounting for, so you should be knowing when they tell you to account for, what do they mean? When they tell you to explain the effect of something, what do they mean? When they tell you to just describe, what do they mean? When they tell you to describe the relationship, what do they mean? Because describing a relationship is a little bit different from describing an effect of something, which is different from just describing a graph directly. So all those ones, you must be comfortable.
Yes. Master,
Yes.
I went ahead. I went ahead. I was trying to read transport in animals, but I got stuck somewhere.
Where?
Hello.
Yes. Yeah, you can tell me where you got stuck and then we see how.
Around the heart. Some
Around the what?
around the heart. If you try to read functional approach, where there is a drawing of a heart, the heart.
Mm. Mm. Yes,
I'm
I can hear
reading
you.
functional approach,
Mm.
but there is a figure there which looks like a graph. There is a figure there which looks like a graph. I failed to interpret it, which has
Oh,
feeding of the atria and reducing of pressure, something of that kind.
okay. Those things of ventricular systole and diastole, or you
If
can
you
call
can
it
throw more light of
diastole.
it on that thing.
Okay. Okay, I'm going to talk about it. Now, I
Yes,
don't know how far you had read,
yes,
but with function approach, actually, it normally has those small brief summary of or about each graph that is normally drawn. Do you
yes,
have that book with you there?
yes, master.
Yeah. Now,
But it is in hard copy.
yeah, that is very okay. Have you opened that page right now?
Yes,
And are you able to see those words, small words, summaries of what the graph is about?
let me read them for you.
Because it will have different scenarios depending
Yes,
on the machine. There is a phonograph.
yes.
Does it have a phonogram?
Mm, I
Hmm?
read them.
Yes. Okay, read them for me.
It's figure 11.11. It says graphs illustrating the pressure and volume changes that occur during the mammalian cardiac cycle. Pressure changes were measured in the left atrium and ventricle.
Okay, okay. You may first pause.
Yes.
Let me open there.
Page 171.
171.
Yes.
Sorry. I'll be on 173. Page 171. This is the one.
Yes.
Okay.
That one.
Yeah. So this is the graph that you've been talking about.
It is above there.
Okay. Just a moment. I want to. Okay, now this is what I've been talking about, that it has what we call a phonocardiogram. This one is going to be measuring sound or pulse.
Yes.
Then you're having electrocardiogram, which is going to measure electrical activity. If you still remember those things of transmission of an impulse, those things, then it also has these different graphs. And for any graph, if you want to understand it, always first of all, read the small subtitles. Now here there is a scale which looks at atrial ventricular systole, and diastole. I don't know whether you understand the meaning of the word systole and diastole. Do you understand what they mean? Hilary, are you there?
No, I'm there. I'm here.
Okay, what I'm asking is that do you understand the meaning of the word systole and diastole?
What I've read, systole is the relaxation and diastole is the I'm forgetting the word. It's contraction, is it? Systole is contraction of the heart.
Okay. Do you know the parts of the heart which undergo systole?
Not yet.
What about the parts of the heart which undergo diastole?
There is the left ventricle.
Okay. Now, what you must know is that
yes,
both the atria and ventricle will undergo both scenarios of contracting as well as relaxing. That is number one. When they are contracting, that's so depending on which part is contracting. So when it is the atria that are contracting, that is atriosystole.
Yes.
When it is the ventricles contracting, that is ventricular systole. Okay.
Yes,
now it should be noted that when for example, when the atria are relaxed, then volume, blood is going to flow into them. Okay,
yes.
let me first get a point here. I'm assuming this is the heart. I'm just drawing a sketch.
Yes.
To show just the major arteries. Okay. Now, I don't know whether you kind of see what I've drawn.
Yes.
Let's wait. Let me get this eraser.
They're supposed to be four.
Yeah. So here, blood is going to be coming from where?
From the parts of the body.
Via the vena cava. Okay.
Yes.
And at the same time, blood is going to be coming from?
No, this is the aorta.
Which one? This one?
No, the other side, the black arrow where it is. No, it is the pulmonary artery.
It is the pulmonary artery which is bringing in blood from the lungs.
Yes.
So from lungs, and then this is from?
From body parts.
Yeah, from body or from the systemic circulation. Okay.
Yes.
Now these of course, these they have valves inside here.
Yes.
Wait. Okay, I've removed it. The pen. Now, what you must know is something is always going to happen here. That blood, of course, we have 4 chambers.
Yes.
Okay. So we have 4 chambers. So blood is going to, first of all, come in, one from the lungs and another from the body at the same time. That is what normally disturbs people. So blood is going to flow into this gonna be the right atrium. This is gonna be the left atrium. This is the left atrium. Yes. Now blood is flowing in at the same time. So for blood to flow into the left and right atrium, these atria must be relaxed. So they are under atriosystole, diastole.
Yes.
When they are relaxed. Now, when they are relaxed, blood volume is going to increase in these chambers. So volume is going to increase. Now, in the process of relaxing, the pressure is low. So as blood keeps on building in, then pressure begins building up here. Okay?
Yes. Yes.
And then eventually they contract. So these, when they contract, the blood is pushed to the lower chambers, that is the right ventricle, and then
the left
ventricle. You get it? As blood, or when these valves open, bicuspid and tricuspid valves, when they open, or you could call them the mitral valves.
Yes.
So when they open, blood flows here. Now, as blood is flowing here, this ventricle is relaxed, it is under diastole.
Yes.
Okay. And then it begins filling. So blood volume keeps on increasing and increasing. So afterwards, these ones contract as these ones are relaxed. So when they contract, they push the blood. Some is going to go through the aorta, and then some is going to go through the pulmonary vein, the lungs. To the lungs. This is the rest of the body parts. Okay. Now, as blood is being pushed to go to the lungs and then to the rest of the body parts, these valves, the mitral valves, close. That is atrioventricular. No, these are the, you can call them atrioventricular valves, or you can call them the bicuspid and the tricuspid valves. They close. Okay, and then blood flows to these 4 chambers. Now, as blood moves, when after this one is contracting, now as the ventricle are relaxing, they may pause what you call, blood may want to flow back. Blood may want to flow back to these chambers. That's where the semilunar valves come in, and then they also do what? They close. When they close, that's what makes that big sound, the dub sound, da. When these valves close, that's what makes the lab sound. This is the dub sound. So that's what they call the lab dub sound that normally happens. Now, I don't know whether you've got that entire flow. So when you have that entire flow, then you can now begin relating it to this graph. Have you understood previously what I've talked about? Now the guy is out. Bernard, are you there? Admin Bernard. Mr. Kakura, are you there? Mr. Kakura, I don't know whether we should end the lesson. We have one student and the network is disturbing. Okay. And I've not heard what you said, but okay. I was still telling Hilary that okay. Yes. Hilary, the network is disturbing, but I don't know what we should do. Okay, in summary, did you get what I just explained previously?
Yes, I did.
Yeah. So, so you relate that information to this. When you look at, you're supposed to know when the sound was made, that is the dub sound and the lab sound.
Yes.
When the dub sound was made, which valves are closing? You see that? And what makes those valves to close? That is what you must know. So, and then the ventricles, you must be knowing. When those valves are closing, you remember that normally for those valves to close, it is the ventricles that have just contracted. So do you expect volume in the ventricles to be high or to be low or to begin decreasing like that? So also you ask yourself, when that sound is made, what is happening in the aorta? You see that? That is in the, what is happening in the aorta, what is happening in the atria, and what is happening in the ventricle. So for you to understand that graph, you must first understand, you should first of all read about atriodiastole and systole and understand them in relation to the heart. Then that's when you can relate the pressures that are occurring in the ventricle at that particular time, and the pressures that are happening in the atria and the ventricle. And then when these valves close, because they all be giving you a clue. Have you understood?
Yes, master.
Okay, I think today we can end there, unless you have any other question.
No, I don't have.
Okay, let's end there. Goodbye and have a lovely day.
Master, will you be posting new videos via YouTube?
Posting new videos. Yeah, I think, we shall be posting. I'll post some videos.
Okay, thank you. What of this work?
This work is already there in Google Classroom. Didn't you get it?
Okay, let me check there.
Okay.
Thank you.
Okay, nice day.
Thank you.